So when it comes to the place of the TCA cycle, obviously it is going to be the mitochondria. Link reaction and the TCA cycle occurs inside the mitochondria. The second thing that we need to remember is that which is the molecule which is going to kick-start the TCA cycle. The molecule which you have made from the link reaction, that is the molecule that you have made as a product of the link reaction, that was acetyl-CoA, is going to kick-start the TCA cycle. So I can say acetyl-CoA is going to start the acetyl-CoA is going to start the TCA cycle. TCA cycle is also referred as the Krebs cycle. Tricarboxylic acid or the Krebs cycle, that is going to occur in the mitochondria, and who is going to kick start it? It is going to be started by the acetyl-CoA, which is of 2 carbons. Now to remember the Krebs cycle, to remember the various intermediate of the Krebs cycle, what are the clinical correlations that we need to understand? So for that we need to understand the carbon numberings very well. If we understand the carbon numbering, then it is going to be easy to remember the various intermediate of the Krebs cycle. So let's see how we are going to remember the carbon numbers. Then we are going to see that how we are going to remember the various intermediate of the Krebs cycle. So let's see that first. So what are the various carbon numbers that we need to remember? See, till now what we know, this is the TCA cycle. Now everyone pay attention, see, just try to understand the carbon numbers. We'll write down this everything, but first let's understand the various carbon numbers, how to remember the carbon numbers. What we know till now is this acetyl-CoA is a 2 carbon molecule, and that is going to kick-start the TCA cycle. This is one thing that we know. Then the thing that I want you to remember is about this molecule alpha-ketoglutarate. See, this alpha-ketoglutarate is a 5 carbon molecule. This is the only carbon number that I want you to remember in this entire Krebs cycle. Alpha-ketoglutarate 5 carbon. This is the only thing that you need to remember. Alpha-ketoglutarate 5 carbon. Now, see here, when the alpha-ketoglutarate goes into the next reaction, what happens is there is 1 carbon dioxide is liberated out. You take out 1 carbon dioxide. 5 carbon, 1 carbon dioxide is out. So how many carbons will be here? 4. Then there is no carbon dioxide is going in or coming out. So all the intermediate are of 4 carbon. Alpha-ketoglutarate is 5 carbon. Down the line, everything is 4 carbon. Down the line from the alpha-ketoglutarate, everything is 4 carbon. Now, this 4 carbon oxaloacetate unites with the 2 carbon acetyl-CoA and makes the citrate. So how many carbons will be there? 4 plus 2 comes out to be 6 carbon. Citrate 6, then the isocitrate 6. Now, when the isocitrate converts into alpha-ketoglutarate, how many carbons? We know it is 5 carbon. So 6 carbon converting into 5 carbon means you have to take out what? Carbon dioxide, right? So means what? Alpha-ketoglutarate is 5 carbon. 1 carbon dioxide is liberated after alpha-ketoglutarate and 1 is before alpha-ketoglutarate. Carbon dioxide, 1 after alpha-ketoglutarate, 1 carbon dioxide is before alpha-ketoglutarate. Alpha-ketoglutarate 5 carbon, down the line everything is 4 carbon, upstream everything is going to be 6 carbon. This is how we are going to remember. This is one point. Now let's see how we are going to remember the various intermediate of the Krebs cycle. So if I ask you that citrate, just answer this question. Citrate is the first product of the Krebs cycle or the first substrate? Citrate is the first product or the first substrate? What you will say? Citrate is the first product or the first substrate? Yes. Yes, it is the first substrate or the first product. Yes, it is the first product. Now, I am giving you a mnemonic like this. Citrate. Now, everyone just pay attention. Citrate is Krebs starting substrate for making oxaloacetate. See, although citrate is first product, but for mnemonic, what I am saying is citrate is Krebs starting substrate for making oxaloacetate, right? So by this mnemonic, we are going to remember the various intermediate also. So now we know the carbon numbers, we know the various intermediate also. So let's write down the reaction part. So I can say that the acetyl-CoA which we have made from the link reaction is going to start the TCA cycle. This acetyl-CoA, which is a 2-carbon molecule, it is going to join with the oxaloacetate. Oxaloacetate is a 4-carbon molecule. They are going to join and they are going to make a molecule that is with the name of citrate, which is a 6-carbon molecule. 6-carbon molecule. Because we are synthesizing a molecule without using any ATP, so the name of enzyme will be synthase or synthetase. As we have discussed earlier also, you are synthesizing a molecule without energy, it is going to be synthase. So the name of enzyme is citrate synthase. The name of the enzyme is citrate synthase. This is the step number 1. Step number 1. Now, step number 2, what we do is, from the citrate, we take out the water. If we take out the water, this is called as dehydration. If we take out the water, that is called as the dehydration process. Now, once we have taken out the water, then what we do is we give back this water again to the citrate. That is called as rehydration. So basically, we are doing dehydration followed by rehydration. We take out the water, then again we give back the water. This 2-step process will convert citrate into a similar molecule that is called as isocitrate. Isocitrate, which is also of 6 carbon. Isocitrate. For this, the name of the enzyme is aconitase. So with the help of aconitase, the citrate will convert into isocitrate. Then this isocitrate is going to convert into the key molecule of the Krebs cycle, that is the alpha ketoglutarate. Alpha ketoglutarate, which is of 5 carbon. We know that alpha ketoglutarate is a 5-carbon molecule. So isocitrate is going to convert into alpha ketoglutarate. In this reaction, what happens is that 6 carbon converting into 5 carbon means you need to take out 1 carbon dioxide. In this reaction also, NAD converts into NADH. Means we are going to generate the NADH. So whenever there is NADH, the name of the enzyme is dehydrogenase. So the name of enzyme here is isocitrate dehydrogenase. This is isocitrate dehydrogenase. This isocitrate dehydrogenase is the rate limiting step of the Krebs cycle. The rate limiting enzyme of the Krebs cycle. So this is how the isocitrate will convert into alpha ketoglutarate. Then this alpha ketoglutarate is going to convert into something called as succinyl-CoA. Succinyl-CoA. We know that alpha ketoglutarate is 5 carbon, down the line everything is 4 carbon. So succinyl-CoA is a 4 carbon molecule. 5 carbon converting into 4 carbon means we need to remove 1 carbon dioxide. In this reaction also, NAD converts into NADH. NAD converts into NADH. So the name of the enzyme is because NAD converting into NADH, so the name of enzyme is dehydrogenase. So it is alpha ketoglutarate dehydrogenase. Alpha ketoglutarate dehydrogenase, and it is also a complex enzyme. So the coenzymes are vitamin B1, B2, B3, B5 and lipomide. B1, 2, 3, 5 and lipomide. With the help of this, the alpha ketoglutarate will convert into succinyl-CoA. So this is how the first 4 reactions are going to take place. So I will just give the numbers. This is the step number 1. Then the step number 2. The isocitrate dehydrogenase was the step number 3. And here is the step number 4. Step number 4. Then this succinyl-CoA is going to convert into succinate. And we know that down the line everything is going to be 4 carbon only. In this reaction, the ADP comes into the picture. We take one phosphate from the succinyl-CoA and converts ADP into ATP. Whenever we transfer the phosphate group, ATP is coming into the picture, so it is going to be a kinase reaction. The name of the enzyme is succinyl-thiokinase. Succinyl-thiokinase. Then this succinate will convert into fumarate. Succinate converts into fumarate. In this reaction, FAD comes into picture and converts into FADH. We know that when there is a NAD/NADH or FAD/FADH is going to be dehydrogenase, so it is going to be succinate dehydrogenase. Succinate dehydrogenase. So that is going to be the step number 6. This is the step number 5. Then lastly, this fumarate will convert into something called as malate. Fumarate is 4 carbon, malate is also 4 carbon. So the name of enzyme is fumarase. And then this malate will convert into oxaloacetate, and in this reaction NAD comes into action and converts into NADH. So that will be the step number 8. The name of the enzyme is malate dehydrogenase. NADH is coming, so it is malate dehydrogenase. So this is how the Krebs cycle looks like. What we have remembered till now, let's revise that first, then we'll see the clinical implication of that. See, we have understood that alpha ketoglutarate is a 5 carbon molecule, down the line everything is 4, and upstream everything is 6. Then the intermediate that we have remembered by this mnemonic. We'll write down this mnemonic also. See here, the mnemonic is citrate. Citrate is Krebs starting substrate for making oxaloacetate. Right? This is the mnemonic for the intermediate. So let's write down this mnemonic first. How to remember the various intermediate of the Krebs cycle. So the mnemonic is citrate is Krebs starting substrate. Citrate is Krebs starting substrate for making oxaloacetate. This is the mnemonic that we are going to follow. Once this point is clear, let's see what are the points that they ask from the Krebs cycle. Now we know the carbon numbers, we also know the various intermediates also. Now we are going to see that what are the various points that we should be familiar with from when it comes to the Krebs cycle. So the first thing that we should be very familiar is what is the rate limiting step. The rate limiting step is the isocitrate dehydrogenase. Isocitrate dehydrogenase is the rate limiting step. When it comes to the energetics part, let's see how much energy we are generating. So to understand the energy, let's bifurcate the energy into 3 parts. NADH produced, then the FADH produced, and lastly is the ATP produced. Then we are going to sum them all. So I can say net gain from one TCA cycle. Right? So how much NADH we have produced? How much NADH we have produced? So when it comes to NADH, we can say that we have got one NADH in the step number 3. Then we have got one in the step number 4, and one we have got in the step number 8. So basically we have got 3 molecules of NADH. Step 3, that is the isocitrate dehydrogenase. Then the step number 4, alpha ketoglutarate dehydrogenase, and lastly is the malate dehydrogenase. When it comes to the FADH, we have got only one FADH. That is in the step number 6. And when it comes to ATP, we have got only one ATP, that is in the step number 5. One ATP only in the step number 5. So let's convert everything into ATP. We know that 1 NADH is equal to 2.5 ATP. So it is 3 into 2.5 comes out to be 7.5 ATP. FADH is equal to 1 FADH is equal to 1.5 ATP. 1 FADH is equal to 1.5 ATP. So comes out to be 1.5 ATP, and 1 ATP is 1 ATP. So we are going to sum them all. 7.5 plus 1.5 plus 1 comes out to be 10 ATP. Comes out to be 10 ATP. 7.5 plus 1.5 plus 10 comes out to be 10 ATP. So net gain. So I can say that the net gain from the TCA cycle, one TCA cycle is going to be 10 ATP. One TCA cycle is going to give us the 10 ATPs. One TCA cycle is going to give us the 10 ATPs. This is the energetics part. Let's see what are the other points, what are the clinically relevant points that we should know. Then comes the inhibitors of the TCA cycle. What are the various toxins that can inhibit the TCA cycles? So that is referred as the inhibitors of TCA cycle. Inhibitors of TCA cycle. There are 3 toxins which can inhibit the TCA cycle. The first is the fluoroacetate, then is the arsenite, and last is the malonate. These are the 3 toxins that can inhibit the TCA cycle. Fluoroacetate is going to inhibit aconitase, that is there in the step number 2. Fluoroacetate inhibits aconitase. Arsenite inhibits alpha-ketoglutarate dehydrogenase, that is there in the step number 4. And the malonate is going to inhibit succinate dehydrogenase. Succinate dehydrogenase, that is there in the step number 6. Succinate dehydrogenase. So these are the 3 toxins which can inhibit the TCA cycle, and out of these 3, the most important is the malonate. Malonate is going to inhibit which enzyme? Malonate is going to inhibit succinate dehydrogenase. Malonate is going to inhibit succinate dehydrogenase. Then the next point that we need to remember is the substrate level phosphorylation, SLP. Substrate level phosphorylation. When it comes to the substrate level phosphorylation, where the substrate level phosphorylation is going to occur, see, I told you that substrate level phosphorylation, when we have discussed in the glycolysis video, we have discussed in that session that substrate level phosphorylation means whenever we are going to generate the energy directly in the form of ATP. See, we can generate the energy in the form of NADH, FADH or ATP. If you are directly generating in the form of ATP, that is called as SLP. So, that is going to occur only once in the Krebs cycle. So, substrate level phosphorylation in TCA cycle occurs only once, and the enzyme for that particular reaction is succinyl-CoA. Succinyl-CoA is an enzyme which is going to do the substrate level phosphorylation. And if they ask that what is the name of the substrate for this particular reaction, what is the name of the substrate? The name of the substrate was succinyl-CoA. Succinyl-CoA is a substrate which is going to lead to substrate level phosphorylation in the Krebs cycle. There is only one place where you are going to generate the ATP directly. Then the next point that we need to understand and we need to remember is that, see, mitochondria is a double membrane structure. Let's say this is mitochondria, and we know that the Krebs cycle is occurring inside the mitochondria, and the mitochondria is a double membrane structure. Let's say these are the outer and these are the inner membrane of the mitochondria. Outer and the inner membrane of the mitochondria. This particular part of the mitochondria is referred as the mitochondrial matrix. This is the mitochondrial matrix. So what we can appreciate is that the Krebs cycle is occurring in the mitochondrial matrix. The TCA cycle is occurring in the mitochondrial matrix. The enzyme of the TCA cycle should be present here. The TCA cycle enzyme should be there in the mitochondrial matrix. But there is one enzyme of the TCA cycle which is not there in the matrix. Instead, it is found on the inner mitochondrial membrane. And this is what they ask, that which enzyme of the TCA cycle is not there in the matrix, instead it is found on the mitochondrial inner membrane. It is not found in the mitochondrial matrix but found on the inner membrane of the mitochondria. So let's write down this point that all the enzymes of TCA cycle, all the enzyme of TCA cycle are found in mitochondrial matrix except succinate dehydrogenase, except succinate dehydrogenase, which is found on inner mitochondrial membrane, Rest all are found on the in the mitochondrial matrix. The only enzyme which is found on the inner mitochondrial membrane of the Krebs cycle is the succinate dehydrogenase. Then the last point in the TCA cycle that we need to remember, the last point to be remembered is, see, there is one enzyme that we have written in the Krebs cycle with the name of alpha-ketoglutarate dehydrogenase. The enzyme of TCA cycle in the step number 4 that we have written was alpha-ketoglutarate dehydrogenase. The coenzymes that you have written for that was vitamin B1, B2, B3, B5 and lipomide. These are the coenzymes that we have written for the alpha-ketoglutarate dehydrogenase. This is in the TCA cycle. Now, when we were discussing the link reaction that was just prior to the TCA cycle, in the link reaction, the name of the enzyme was pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex. In the pyruvate dehydrogenase complex, the coenzymes were vitamin B1, B2, B3, B5 and lipomide. Means which is exactly same as alpha-ketoglutarate dehydrogenase. That is exactly same as alpha-ketoglutarate dehydrogenase. You can see that they are having the same coenzymes. 1, 2, 3, 5 lipomide, 1, 2, 3, 5 lipomide. So what they ask is that which enzyme of the Krebs cycle is same as pyruvate dehydrogenase complex. Which enzyme is same as pyruvate dehydrogenase complex in the Krebs cycle? The answer will be alpha-ketoglutarate dehydrogenase. So let's write down this point that alpha-ketoglutarate dehydrogenase is an analog of, analog means similar, is an analog of pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex, that is called as PDH complex. So this is the point to be remembered, that which enzyme is an analog of the PDH complex. The answer is alpha-ketoglutarate dehydrogenase. Okay, so these are the points to be remembered for the TCA cycle. Now, because we have discussed the glycolysis, we have discussed the link reaction, the Krebs cycle all. So let's take a example, a practical example. We can say that we are having one molecule of glucose, and we have all the enzymes available. We have all the things available. We have oxygen available. We have mitochondria available. We have everything. And we have this one molecule of glucose. How much ATP we can produce out of that? Yes. So, let's calculate how much ATP we can get from one molecule of glucose. So, first of all, this glucose is going to convert into pyruvate. When the glucose converts into pyruvate, this is called as glycolysis. And glycolysis is going to give us how much ATP? 7 ATPs. Glycolysis gives us 7 ATP. Then this pyruvate is going to go into the link reaction and will convert into acetyl-CoA. Is going to convert into acetyl-CoA, and this is called as link reaction. When pyruvate converts into acetyl-CoA, in the link reaction we have written that NAD converts into NADH. NAD converts into NADH, right? So NADH is equal to how much ATP? 2.5 ATP. 1 NADH is equal to 2.5 ATP. Then this acetyl-CoA is going to kick-start the TCA cycle, and it will do the TCA cycle. One TCA cycle gives us how much ATP? Just now we have calculated. It is going to give us 10 ATPs. One TCA cycle gives us 10 ATP. So, how much ATP we are going to get? See, when you do the glycolysis, my question is, how much pyruvate molecules you are going to get? How much pyruvate you are going to get? You are going to get 2 molecules of pyruvate. So, how many times this link reaction is going to happen? This link reaction is going to happen for 2 times. So, you are going to get 2 molecules of NADH. 2 molecules of NADH means comes out to be 5 ATP. 2 into 2.5 comes out to 5 ATP. 2 acetyl-CoA is going to run the TCA cycle for how many times? 2 times. 1 TCA cycle is 10 ATP. So 2 TCA cycle will be 20 ATPs. 2 TCA cycle will be 20 ATPs. So these are the energies that you are going to get. So let's sum them. 7 from the glycolysis, 5 ATPs from the link reaction, 20 ATPs from the Krebs cycles. So 20 plus 7 plus 5 comes out to be 32 ATPs. So let's write down this point that 1 molecule of glucose via complete aerobic oxidation will provide 32 ATPs. 1 molecule of glucose via complete aerobic oxidation will provide 32 ATPs. So this is what we need to remember regarding the TCA cycle. Yes, these are the things to be remembered. 1 molecule of glucose, after all these things, are going to give us 32 ATPs. Yes, thank you guys.